Hello, my name is Christian, and I want to thank the organizers for giving me the opportunity to present some of our work we do in the field of systems biology and the role of Perl in this field. I would like to start with a little introduction of the company I work for. So it's a non-profit organization, it's called ASIP. It stands for Austrian Center of Industrial Biotechnology. We have approximately 200 employees. Many of them are scientists or PhD students, and I work for the Metabolic modeling group there. So I'm working on simulation of metabolic activities in living cells. I'm also an associated member of the University of Natural Resources and Life Sciences in Vienna. It's also called BOKU, at the Institute of Applied Microbiology, and I'm also an associated member of the Royal College of Surgeons in Ireland in Dublin. And luckily I also enjoyed some formal education. I did a bachelor in computer science, a master in electrical engineering, and a PhD in computational physics. From there I drifted into biophysics and ended up in systems biology. This is the outline of my today's talk, so 3 parts as you can see. The first part is about growing bone artificially. In particular there I'm talking about simulating flow perfusion bioreactors. Part B is about using living cells to produce substances of interest. Here I'm in very funny. I didn't mean that. So there I'm working in identifying the best gene intervention strategies to optimize the productivity of those cells. And part 3 is about lab-on-a-chip systems, and here I'm focusing on simulating and optimizing dielectric cell detection sensors. So I must say that you won't find a lot of Perl code in this talk, so I hope you're not too disappointed if there are only a couple of lines of Perl code in there. I'm more focusing on the general concepts and the process I had to do. So if I see that many of you fall asleep, I will immediately stop my presentation and hand over the microphone to the next speaker. So let's start with presentation part A. So bone tissue engineering, the field is called. So why do we need the field of bone tissue engineering in the first place? So there are 3 main reasons why human can suffer from bone loss. Number 1 is cancer, number 2 are usually accidents, and number 3 are tooth extraction. So if you're ever involved in a bar fight or a pub fight, then hopefully your opponent will look like that after a couple of months. So if you lose teeth there, then after a while the bone starts to degrading and disappears because the mechanical load that your teeth are putting on the bone there is missing, and then the bone degrades and disappears after a while. So a few key facts about bone tissue. So bone tissue is a very complex tissue. It consists of organic material like the cells and the inorganic material, the calcium phosphate. Important is that the bone is very well supplied with blood. So we have a lot of vessels in bone tissue. Number 3 here is bone is constantly remodeled, so the human body constantly creates new bone material by cells called osteoblasts, and there's a resorption of bone material all the time, all day long, and old bone material is removed by osteoclasts. And if there's any disbalance in this creation, generation and removal process, usually we get osteoporosis, so which is a very important issue, especially in elderly people. And as I just mentioned before, if there's no mechanical load on the bone, bone loss will happen to you. This, for instance, happens also to astronauts when they are in space, so the missing gravitational forces cause bone loss in them. So this is the reason why tissue engineering or bone tissue engineering is a rather complex issue. Of course, we could do a self-donation of bone. So if you ever need bone material somewhere, you could take it from your hip bone somewhere here and implant it somewhere else. But there are a few unwanted side effects. Number 1, we need another surgery. Often those patients are already sick, so they have cancer, had accidents, so this is stress on them, which we don't want to have. Number two, interestingly enough, this is actually a very painful procedure and is often the cause of long-term problems. So very often the hip is then the problem, not for instance, your mouth anymore. And then with any operation, there's a risk of inflammation. So there's another way to get to bone tissue, and this we call bone tissue engineering. So this is kind of a circle here. So we start with our patient. We take stem cells from them. We only have a limited number of stem cells. Usually they are taken from the bone marrow. Then we multiply those cells. Then we take a scaffold. We seed the cells on the scaffolds, put it into a bioreactor, let it grow there for a couple of weeks, and hopefully after a certain amount of time we can harvest suitable bone tissue that we can implant into our patient. The problem here is that we take the stem cells, and the stem cells, usually from the femur, need a special environment. So stem cells, as you all know probably, are cells that can differentiate in a variety of cell types, like bone, like cartilage, like a lot of other cells we have in our body. Important is that we need to provide these stem cells a proper environment that they survive, so they need nutrition. We have to remove the waste products, the metabolic waste products, and we have to provide the mechanical load. So how do we achieve this? How do we provide this environment? We do this with a bioreactor. If this mechanical load is not in the right range of forces and pressure and shear stresses, then something else will happen. So we don't get our bone material, so we would get, for instance, ligaments or tendons or something else we can't implant there. So as I said, we use a bioreactor to grow our bone. So this kind is the bioreactor. In here we put our cell-seeded scaffold. We have a syringe pump where the nutrition fluid is in. We pump it through this scaffold, so the cells grab the nutrition, produce waste products, they're pumped through. And as you all know, the body only works in a proper way when we deal with, it is at 37 degrees Celsius. So all this has to happen in an incubator. And what I want to achieve was, or what the goal was, I want to do a numerical simulation of this flow process in here to get the perfect bone tissue. There I faced one big problems. So when you put the cells in there and into the scaffold, and for this simulation you need kind of a geometry, and usually you do this with X-rays or micro-CTs, microcomputer tomography. The problem is that the cells do not absorb X-rays. So we can't use the regular way of doing a micro-CD of the cell-seeded scaffold, and here Perl came to the rescue. I want to say two more things about those scaffolds and the cell attachment. Number one, there are many different scaffold types. In the lab where I work, we use two. We had a collagen gag scaffold that had very small pore sizes and was more soft. The other one was a very brittle and hard material with big pores. And these are the key properties, and they are important for one main reason, the cell attachment. So this here should, represent the cell, and cells like to attach to flat or convex surfaces because they're attaching all down here. If the surface is concave, cells tend to lift off, and they're only attached down on this small region. And of course, they completely behave differently depending on this morphology. So we in general deal with two types of cell attachment. The one I called flatly attached cells, and the other one is bridgingly attached. So for that cell proceeding process, I wrote a little Perl script. Actually, it was kind of a Perl environment. So the main part there was object-oriented Perl with a couple of classes, like the scaffold was represented by a class, the cells group, the cells, and here we had two kid objects or classes, the flat cells and the bridging cells. And I heavily used the Monte Carlo approach, so a lot of random number generation, for instance, to determine the location where the cells are put into the scaffold, the orientation in space, how the cells are oriented, and also the morphology was randomly determined. So the general concept of this program looked as followed. So I determined first what type of cell attachment the next cell I attached was. Was it flatly or bridgingly? Then I tried to find a spot on this scaffold where the cell head is going to be attached. Then I was randomly picking a direction, and then I tried to find a spot for the tail, and then I was propagating this cell through the room, through the space there. And of course, if it was a flatly attached cell, it had to propagate along the surface, and if it was a bridging, it had to go through the void. And whenever there was any obstacle or something detected, then the procedure was stopped. So whenever any of those steps failed, I restarted this certain procedure until I could seed a predefined number of cells. So the input for this program was mainly the scaffold geometry, which we got from the microCD. The input then was in a GNU-zipped text file. Then another important parameter is the cell density, so how many cells per whatever volume are we going to seed, and then of the ratio of flatly to bridgingly attached tail, which we determined in advance for a huge set of different scaffolds using confocal microscopy. So the program then looked pretty much like that there was this scaffold object I got with some input data, the cell parameters, and I created an object of the class cell group. Then I started a while loop, and I iterated through this loop until the number of cells that was determined up above there was reached. So first got the cell type, and then I tried to add a cell to the cell group. Whenever this was not possible anymore, because it was already so tightly packed with cells, I stopped the program. Otherwise, if all cells could be seeded, the output file was written, and the output file again was a GNU-zipped text file. This now is a representation of this numerically seeded scaffold. So the red things are the cells, the blue one is the scaffold, and you see here, for instance, such a bridgingly attached cell, and there are definitely also flatly attached cells in there. And this scaffold now was used as input for the fluid dynamics, a CFD simulation, and a so-called elastostatics or deformation simulation of the cell. Most of it was text-based. The visualization of these pictures or geometries was done with a program called ParaView. So what were the steps I had to do during this project? So first I had to do the scan of the scaffold with the microCD, then I had to numerically seed the cells onto the scaffold. This was done with Perl, as we have just seen. Then I created the geometry for the computational fluid dynamics program. The fluid dynamics program was called, or is OpenForm. It's an open source tool. It's actually very good, very fast, and in the CFD community, very popular tool. The main advantage of OpenForm is it uses text file as a command line tool. It uses pure text file for input and output. So of course then Perl comes in, and being so good in handling text files. And then I ran many CFD simulations for different parameters, cell densities, viscosities, and all these CFD runs were automatically controlled using Perl. Then when those runs were finished, post-processing was necessary, so I took the results, got them from the text files, set up the CFD simulation for the deformation simulation, and there I tracked every single of thousands of files. So also this tracking and getting the information was done with Perl. So again, text file for OpenForm. It's a command line tool, runs on Linux. Each CFD case is residing in its own directory. So the main features and commands I used in Perl were make path, copy, of course system for running all those simulations, unlink for cleaning up and removing trees, and a lot of regular expressions were used to extract the information. So what do we get after the CFD simulation was done? We get 2 or 3 main results. So what you see here is the fluid flow through the scaffold. So these white spots here are the scaffold and the cells. And you see that the fluid is going through, and the red areas show you that the velocity is very high, which means that close to the walls the shear stress is very high. And here you see the pressure. Those two things, so the shear stress and the pressure, were used to then simulate the deformation of cells. So they were applied to these cells, and then the mechanical stress and the deformation were simulated. What was the outcome of these simulations? So it was, I think, the first time that someone quantitatively evaluated the stress, the mechanical deformation that was acting on the cells, and we did that for these 2 cell types. And of course, it's kind of obvious, but we now found out with numbers that bridging cells are much more sensitive to the flow. So this means for different scaffold types, we needed different flow rates. And what the outcome was that we could determine better flow velocities than they were used before. So we could reduce the time until a bone could be harvested from around 4 weeks to 2 weeks. And this is really a huge improvement because, first of all, it's a shorter waiting period for the patient until the operation can be done. And secondly, the lab costs are hugely decreased. I must confess here that the bone we grow, other groups grow, is still not perfect. So there is one major issue that is still not solved. This is the generation of vessels. So the vascularization is still not perfect yet. And if you're interested in any details of this work, we have published some papers about that. Conclusion, here Perl was used for every single task except for the computational fluid dynamics simulations and the elastostatic simulations. Those 2 tasks were done with OpenFOAM, this open source software, and this was done on a high performance computer. So this was 5 or 6 years ago. At that time, 250 gigabyte of RAM was really a pretty nice computer. So this was part 1 or part A. Now I come to part B. It's about the cell production hosts, or cell factories we call it, and finding gene intervention strategies that optimize our cells. So what is our goal? So we want to design microorganisms, and these microorganisms should produce substances of interest. So please see, we are mainly interested in ethanol, not for drinking, for other reasons. And they should do that as efficiently as possible, and also important from a given substrate. So for instance, we are interested mainly in glucose, although we are shifting this now to other substrates. So why are we interested in ethanol? Because ethanol can be used as motor fuel. As you all know, it's already now an additive for gasoline, which sometimes is called biofuel. And of course, we all know that fossil fuels are limited. Ethanol is a renewable energy form, and we don't get the problems with the CO2 there. So how do we increase this efficiency? So when a microorganism is producing ethanol, it does it by microbial fermentation of sugar. So what we do, we optimize this metabolic process. What is metabolism? It's the set of chemical reactions that happen in living cell to sustain life. Good. So what do we need for that? We need 4 things. We need a metabolic network. We need elementary flux modes, and we then need or computational minimal cutting sets, which are actually the sets of genes we want to knock out. Those 3 concepts I will explain in a minute. And we also need a computer, Perl, and a linear programming solver. So what is a metabolic network? A metabolic network is nothing else than a set of metabolites and chemical reactions. What are these chemical reactions? They transform the metabolites from one form to another. Here's a little example of a typical metabolic reaction. And these metabolic networks are usually stored in a format called SBML, Systems Biology Markup Language, which is an XML dialect or they're stored as in a stoichiometric matrix. When we talk about those networks, then the final goal is always to deal with the genome scale metabolic network. So there we deal with several thousand metabolites and reactions. So this picture, you can hardly see it, but this is actually a rather complex system, such a metabolic network of a regular cell. So, as I mentioned, we also need elementary flux modes. What are elementary flux modes? You see here a little toy network. So the other one was this big one, genome scale. This is a very small toy network we are usually testing our programs with. These tilted squares are directions. Those rectangles are the metabolites. And an elementary flux mode is nothing else than a pathway through such a network. So metabolite is transferred by reaction 1 to A, by reaction 2 to B, and then to whatever product here, the biomass. And this pathway has to have 3 key features. Number 1, it must be in steady state, which means whatever goes in here has to come out somewhere else. So accumulation of anything in there is not allowed. Number 2, it must be thermodynamically feasible. And number 3, an elementary flux mode is always minimal, meaning if I take away one reaction, the entire pathway collapses because the steady state condition is not obeyed anymore. And for this simple network, for instance, we have 4 elementary flux modes. So why are these elementary flux modes so interesting to us? The main property is that when you compute all those flux modes, you know, in metabolic terms, exactly what your organism, your cell, is capable of biologically. This means whatever operation condition it works in, you always can get into this or describe it by superposing the flux modes we computed. So this is a very simple diagram, a 2D representation of the flux mode. Here we have the biomass. The biomass means it is reproducing, it's growing, it's dividing. And here we have the ethanol that we want to produce. So what we want to have is an organism that is somewhere up here, high biomass, so it's reproducing, regenerating, and high ethanol. We don't like anything here because there's no biomass, it's not growing, it's dying actually. We don't want to have anything down here because it means we don't get any ethanol. And the interesting point now with this superposition is we can put or draw around our elementary flux modes an envelope, and whatever happens, our microorganism is in here metabolically. Why is this important? So if you have a look at this medium-sized model of an E. coli system, then we can compute the modes here, the elementary flux modes, again biomass, again ethanol. So these yellow ones are the nice ones we want to have. So usually what you do when you are optimizing such a microorganism, you start to knock out genes. Knocking out genes meaning you're taking away the enzyme that drives the reaction. And when you do that, many of the modes go away, as I said, because of the minimality constraint. If you knock out one gene, you see only the blue ones survive. But when we draw around the envelope, we see we are still down here. No ethanol might be produced. The same is true for this other candidate of knocking out. But when we knock out both of the reaction, we suddenly see that the blue ones are up here. So down here are no surviving modes. I draw an envelope around here, and I see whatever happens, this organism has to produce ethanol at any cost. So what we came up with then, and where Perl again helped us a lot, we came up with an algorithm that computes the optimal knockout set. So the input for this is we compute the EFMs, put it into our algorithm, and then we tell how many deletions we want to do. The more deletions, the more expensive in experimental respect it is. So for instance, if we just want to do 3 deletions, we get something here, high biomass, high ethanol. More deletions, we are pushing this area up, and our little E. coli is getting better and better. The concept we used here was is called linear programming. I don't know if it's known to you. It's a way to optimize linear objective functions that are subject to a system of linear inequality or quality constraints. So it typically looks like that. We want to optimize something. So these are all vectors here. This is a matrix. So we want to optimize a linear function and have these inequalities here. It originally, this whole linear programming stems from research done in military and economy around 1940s. And what really like about it is that the main contributors, one of them was George Danzig, and he's actually the character Will Hunting from Good Will Hunting is based on. So he once came to a class and he found on the blackboard a couple of, or actually 2 problems, and he was late and he thought this is homework. And actually they were the 2 unsolved big problems in his field. And he didn't know that, so he went home, started doing the homework and realized this is a bit harder as usual, but he managed to do it. Then he went back to his professor and gave it, and he still was thinking this is homework, and after a couple of weeks the professor came back and told him that he has solved those 2 big problems. So much to the trivia about this linear programming. What are the main challenges here? So one main challenge is that this number of elementary flux mode grows exponentially. So this little model, E. coli model I showed you, has a couple of thousand modes, but if we go genome scale, this is just incredibly huge. Even medium scale model have easily a couple of 100 million modes. So we always need high performance computers now. So our computers have 384 gigabytes of RAM currently. And as I said, genome scale models no one can do right now in the world. So what were the steps we did here? And now again, this is the connex to Perl. So we get our SBML files. Those are published by other groups who are more in genetics. We take this, check the consistency of the model, and fix the models. So usually we do that with www.mechanize and access those websites for biological data. For instance, KEGG is, I guess, the most famous one. Then we do a preprocessing of the SBML file, so we create this stoichiometric matrix I mentioned before. So there we use Perl's XML modules, for instance, XML simple. Then the computing of the EFM has to be done. Here we can't use Perl because this is a real high performance issue. For that we use EFM tool. It's in Java, so you might wonder why this is then high performance, but it's still the best tool out there. And after that, we compute the minimal cut sets, and this again is done with Perl. And for that, we use an external linear programming solver, and I wrote a little Perl module for that, which I will talk about in a minute. And then we do the post-processing and visualization of the results, and this is done in Perl plus programs like GNUplot or R. So what is the Cplex? Cplex is a commercial solver implemented by IBM. It's a typical linear programming solver. We have alternatives here. The free one is GLBK, which is unfortunately not very powerful, and there's a commercial one, it's Gurobi. As I said, Cplex is fast, supports multi-threaded execution, and has even more advanced features like quadratic solvers or mixed integer solver. The good thing here with Cplex and Gurobi is there are free academic licenses available. So Cplex comes with a C library to support all the Cplex function. So what I did, I wrote the math Cplex module based on XS to link to this library, and currently math Cplex supports many but not all exotic features of Cplex. And if we have a look at that, I don't know if you can read that. So it's the regular way you include the module. You get the Cplex environment, and you start with an empty linear program by creating a new optimization problem, OP. Then you tell the linear program if we are doing a minimization or maximization. Then we define the variables and the objective function in the next step. And then we add the constraints, the inequality and equality constraints. And we add them by adding rows. And then we can write our linear program to a file. So the LP format is a very common and popular format in linear programming. And then here the real work is done by Cplex optimizing the problem. Then we check the status. Was it optimality was reached or not? Was it infeasible? So there are different states. If everything was fine, we get the solution and print it. And of course, after all that, we have to clean up the environment again. So what did we achieve with this entire project? So we, of course, had to experimentally verify our models, and we took a E. coli strain that was originally published by a group in Minnesota. And they also tried to optimize an E. coli strain for ethanol productivity, and they came up with an approach where they used 8 deletions, so they had to do 8 knockouts. We used exactly the same strain, used our model, and we could find a way where we only needed 5 deletions. And this is actually a huge improvement. If any one of you knows how experimentalists hate to do many knockouts in a single organism, so they really liked us for reducing their workload. And as you can see here, this is the one by Trin, and this is our approach, and they more or less have identical biomass or ethanol production here. Good. Again, we have published some of the work in these papers. A few more words here. First of all, other applications of this approach, so you could optimize your organism for other substances. Nowadays, many of the pharmaceuticals that we can buy or get are made by living cells. So if our model or the metabolic networks covers these other drugs, then we can optimize for them. The other thing we used our program for was computing the optimal combination of cancer drugs. So as you might know, usually if a patient has cancer, then he has different cancer cell types in his body. And for these different cell types, cancer cell types, different drugs are required. And finding the optimal drug combination that kills all of the cancer cells in the body is also such a hitting set problem we used our program for. And another application might be DNA sequencing. So we used our programs to do multi-genome alignment with that. Good. One more note on genetic engineering, as many people are kind of afraid of it. So first of all, when we compute such a knockout set, it doesn't necessarily mean that we have to really knock out a reaction. Sometimes it's good enough to just not provide a substrate, special form of sugar or whatever. The other thing is, whatever we do, we are changing our little microorganisms, and then we put them into a tank, usually a huge tank with hundreds or thousands of liters. And there they live, there they produce ethanol or whatever. And then when we are finished with them, they usually are killed. So the entire broth is heated up, that they are gone. And the rest then is taken care of by purification plants. So nothing is released to the environment, what we do here. Good. As I see, I think I'm completely running out of time. I'm just kind of going through the last part. So this was the third project actually I wanted to present to you. It's about lab-on-a-chip systems. These are little devices that integrate many different laboratory functions into one single chip. So what you see here is usually a couple of millimeters or max centimeters. Very often in here we have living cells. We need nutrition fluid again, and they have some advantages or many advantages. So very fast analysis, less waste products, lower cost of the medium that we supply. So the medium can be very expensive. If you have just microfluidic applications, then this is really nice. Then parallelization. So even on such a chip, we can have many of those experiments in parallel with the same environment. We can do online monitoring. So very often in biology, you have to stop or terminate your experiment, kill the cells to get any data from them, so this is not necessary, and it's very small, so there are many different applications we can think of. So this is what it looks in our lab. So we have here the inlet and the outlet of our fluid. So this is the little chamber. It looks like that. So here the fluid gets in, this is the chamber, fluid gets out, and in this chamber we have living cells that are attached to the surface, and here I did a little simulation of the shear stress. One of my favorite applications of those lab-on-a-chip systems is, for instance, the artificial lung. So we have here a system where we have two microfluidics, air, the lung cells, and blood. All those is provided with the proper environment again. And we can then test, for instance, pharmaceuticals, how they act on those cells, out these lung cells are also toxic materials, and this is all working on these little chips actually. Good. So what we were working on was increasing the dielectric sensor. So this is kind of a finger sensor where we put voltage on it. So this is a cross section. So here we have the electrodes and the electric field, the electric flux enters here the cells. So this is the chamber. Here we have the cells. The fluid goes through here and we detect the cells by the electric field change or the electric change of the capacity, because this is nothing else than a capacitor. And the nice thing is that we can here monitor with a wide range of frequencies. So I'm just skipping through because I'm running out of time. Here you see the flux. So we came up with a new design where we put in here permittivity insulators, that the field really was entering up here into the region where the cells were located and didn't just shortcut through that. And by doing so, and by doing a lot of numerical simulations, again with our CFD program OpenFOAM, as electric flux is just a flow, as thermal flow or a liquid flow, we could optimize them. Here we also were verifying our simulations. Math complex and fitting optimization tools were heavily used. And at the end, we could increase the measurement sensitivity of our sensor by a factor of 2. So this brings me to the conclusion of all 3 parts. So as you might have seen, Perl is a key player in many projects in life sciences and systems topology. Perl is in particular very powerful in pre- and post-processing, in process control, automatization, and we also use it a lot for prototype development, testing new algorithms. Whenever we come into high-performance computing and we really need fast execution, we go to a low-level language, so usually C, C++. Very often also we do kind of a hybrid where we use Perl for the easier, not high-performance critical issues and combine it with C, C++. The question, could we have done this with another language? Yes, of course. Could we have done this with faster or better with another language? Certainly not. So, acknowledgements, and thank you for your attention.